In this video, we'll give an intro to MACS, which is Model-Based Analysis of ChIP-seq. First, we'll give an overview of the model used in MACS, and then go through how to use MACS and analyze the results. So briefly, ChIP-seq is a technique used to identify in vivo binding sites of transcription factors or histone marks. The protein and DNA are cross-linked to covalently link the complexes, and then the DNA is fragmented by sonication. Then you pull down your transcription factor with an antibody, and the bound DNA is also pulled down. You can then reverse the crosslink and sequence the bound DNA. This generates millions of sequencing reads, but then how do we identify the bound sites? The first issue is the type of reads that we'll get. So the DNA is randomly shared into fragments. On average, the transcription factor will be bound towards the middle of the fragment, but the DNA sequencing reactions will happen from either end of the DNA fragment. So you'll end up with a bimodal distribution of reads. MACS handles this by identifying 1,000 high-confidence peak regions where there are many reads, and identifying the average distance between the two peaks, which is d. It then goes through all the peaks and shifts the reads towards the center by d/2. So the result is a unimodal distribution of reads centered where the TF is actually bound. So to summarize, MACS models the shift size by looking at 1,000 high-confidence peaks, then shifting all of the reads. This modeling must be done for each transcription factor because transcription factors will have different sizes of d. Another issue is how to identify significant peaks. This is done by comparing the read distribution to that of a control sample. A Poisson distribution can model the read distribution with the parameter lambda representing the background frequency of tags. A 2D long window is slid across the genome, and windows significantly enriched in reads over the background, ChIP peaks, are identified. However, this method can give false positives. This is because in the background sample there are local coverage biases due to chromatin structure, sequencing biases, or copy number variation. So instead of using one lambda across the entire genome, each of the putative peaks is reanalyzed against the Poisson distribution with a local lambda. The lambda local is the maximum of the original lambda, or lambda 1k, 5k, or 10k. These are local read frequencies in windows centered around the putative peak location in the control sample. Peaks are called significant if they have a p-value less than 10 to minus 5 against this local Poisson distribution. The fold enrichment for each peak is defined as the tag count in the window divided by lambda local. The false discovery rate in MACS is defined empirically. ChIP peaks are found by looking at enrichment over control. Using the same parameters defined there, peaks in the control over the ChIP sample are called. These should all be false peaks because they're called from the control sample. The false discovery rate is then defined as the number of control peaks divided by the peaks in the sample.
Okay, so to do a ChIP-seq analysis, first of all, you should get your reads mapped to the genome, and there are plenty of tools that can help you do that. For example, Bowtie is one of them, and it is pretty much widely used these days. And after you get the result from the mapping, we can now get into the MACS. And MACS is the tool that will help you to call the peaks. And with the output files of MACS, we can do several following analyses. So in the following slides, we're going to cover 3 important aspects of MACS implement. And so they are command lines with key parameters that you should specify, and concerns about the narrow versus broad peaks, and also we'll talk about a little bit about the output files format. Okay, so one thing to keep in mind that is when you map your reads to the genome, you have to specify that there will be only one hit on the genome per read. So to do this, we use m-1 in the command line, and also -S is used to indicate proper output format. So here -S means the output format should be the SAM format. So then we will introduce how to implement the MACS to call the ChIP-seq peaks. And the basic structure is like the first line shown here. In the example command, the letter F indicates the format of the tag file, and the T specifies the path and file name of chip-seq data, and the dash small c specifies the control data, if you have them. And the parameter -g gives the genome size, and here HS is the shortcut for human. And the prefix of output files is set via the command -n, and capital B or S indicate the outputs you want. So this is a table that summarizes some important parameters you could use while calling MACS. So you could refer to this if you have specific questions of yourself. So ChIP-seq is commonly used to analyze transcriptional factor binding site or histone modification distributions. However, because these two categories give us very different shape of signals, so we should make some modifications in the command lines. Transcriptional factors often give like sharp and discrete signals, but some histone modifications have much broader signals, and thus make the shift sizing model building very difficult. The most common histone modification with like broader peaks include H3K27ME3, H3K36ME3, and H3K9ME3. To skip the default setting of the shifting size model, we should include the command -no model in the line. So if you want to adjust the default size, which is 100 BP in the no model setting, you could specify the shift size here. Furthermore, if no control is available, the local background estimation should be skipped. And to do this, you should add no lambda in the line. So here's an example. In them, you can see how to use the features no model and no lambda while you calling MACS. So after the hardworking computer finishes the MACS, we could take a look at what output files we get. There you should have a R script for you to produce a PDF file containing a shifting size model. And you could also get an XLS and a BET file, which contain the peak information. And if you want to visualize the peak distribution, there's this BET graph file you could use. So that is the implement part of MACS. In the following part, there will be demo showing you the analysis you could do with the output files from MACS.
Four main output files of interest for analysis. The first is the peaks.xls file, which contains information about called peaks that you can sort or filter using Excel functions. Next is the peaks.bed file, which contains the peak locations. This is especially useful for further analysis using the UCSC genome browser or visualization with IGV, which we will demonstrate later. The summits.bed file contains peak summits locations that are useful for downstream motif identification and analysis. Finally, the model.r file is an R script that will produce a PDF image about the model based on your data. We'll start with the explanation of the model.r script and then move on to IGV analysis. The model.r script generates a PDF file containing an image of the model based on your dataset. To generate the model, load the script into R using the command shown above. Using RStudio generally will not work because the size of the data may be too large, so you should run it from the command line. This is an example of the image generated using the model.r script. As you can see, it will display the percentage of forward, reverse, and shifted tags in relation to their distance from the midpoint of the paired peaks window. The red curve is the distribution of peaks from forward tags, and the blue is the distribution of peaks from the reverse tags. The black curve is the merged distribution of the shifted forward and reverse tags. The magnitude of the shift is determined by the distance between the summit of the red and blue curves divided by 2. Next, we will show you how to visualize MACS with the Integrative Genomics Viewer, which can be downloaded from the Broad Institute website above. So here's the Integrative Genomics Viewer website, and you can download the program under the downloads link here. There's the option to run IGV through the website by launching the Java Web Start application, but this may not work on some versions of Apple due to security problems. So to run it off your computer or on your server, you can download the binary distribution here. So once you download the IGV program, you unzip it and you can see that there's a lot of files, but all you need to do is click on igv.jar to launch the application. So this is the Integrative Genomics Viewer, and it comes preloaded with human HG18, but if you want other genomes, you'll have to download it from the server, and you can see there's a lot of different types, human, mouse is the one we want, but I've already preloaded that one, so just click on that tab and choose mouse. You can see the RefSeq genes change down here to the mouse genome. And we'll load our file, and you can do that from the server, the URL, but I just saved it to my desktop. And this one will be the peaks.bed file. And here you can see that on the macro level you can't really see the max peaks, but if you click on the chromosome there, you can see the RefSeq genes, but once again you can't see that because we only mapped chromosome 7 in this case. When we change it to that, you can see the annotation, which will give you the score and the chromosome position, and also the gene loci. A lot of features to explore, but since this is a basic introduction, we'll stop here.